ये एक साइट का नाम क्या है आपकी उसमें यूट्यूब पे?
अस्सलाम वालेकुम सर, हमारे चैनल का नाम है रावेलियन स्टूडेंट्स रिसर्च सोसाइटी।
क्या, सॉरी, क्या लिखा हुआ है बेटे?
RAWALIN
AWALIN,
N
AN
यस
नहीं, LIN।
LIAN LIAN
राइट, फिर?
स्पेस स्टूडेंट्स
अच्छा, स्पेस भी है इसके बाद? स्पेस स्टूडेंट्स रिसर्च।
स्पेस रिसर्च सोसाइटी। और मैं आपको लिंक भी भिजवा देती हूँ, ये बाद आपके साथ शेयर कर देते हैं।
हाँ, लोग मुझसे पूछ रहे हैं डायरेक्ट, मुझे तो कुछ नहीं पता किधर क्या है। आप भेज दें मुझे, मेरे व्हाट्सएप का नंबर पे भेजना पड़ेगा, 0331 817 435।
सर मैंने भेज दिया, मैंने
भेज
लिंक
दी?
भेज दिया।
ओके, ग्रेट, थैंक यू सो मच। एक्सीलेंट। कुछ लोग आए हैं उसमें बेटे, यूट्यूब पे?
यस सर।
चलें, फिर आप लोग बता दें जब भी, मेरे पास तो टाइम है, आप लोग देख लें कितना, कब से शुरू करना चाहें।
सर विल वेट फॉर अ फ्यू मोर मिनट्स, अभी 50 पीपल आ गए हैं तो बस थोड़ा सा वेट करेंगे फिर स्टार्ट
अच्छा, मुझे नजर आ रहा है ये लाइव चैट 50 करके, अच्छा दैट्स वेयर,
यस
ओके, ठीक है, alright, ठीक है। आप लोग सारे RMU से हैं?
यस सर।
ठीक है, कौन से ईयर से, फाइनल ईयर?
यस सर।
ओके, एक्सीलेंट। कुछ लोग आए हुए ऑनलाइन, उनको कुछ सलाम दुआ कर दें, अदरवाइज दे आर थिंकिंग के वी आर क्वाइट.
Okay, sir, we are starting.
ठीक है, बिस्मिल्लाह.
Isaac, you're mute.
Assalamualaikum everyone. Good evening. I'm Isaac Bal from final year MBBS, member of Research Development RSRS. I'm really glad to have all of you with us today. The ABCs of Randomized Control Trial is a 3-day course which covers everything from basic introduction to the statistical analysis of RCTs. Before we start formally, I'd like to invite Fatma Faraz, President RSRS, for a few words. Fatma?
Thank you so much, Aiza. Good evening and Assalamu alaikum everyone. I am Fatima Faraz, President RSRS, and I'm glad to have all of you here today. Randomized control trials are considered the gold standard in clinical research, and they provide the highest level of evidence for evaluating the efficacy and safety of interventions. And it is my utmost pleasure and honor to kickstart this event dedicated to exploring the randomized control trials. And before we all delve into the world of evidence-based medicine, I'd like to take this opportunity to invite all of you to learn something that's even more superior to randomized control trials in the hierarchy of evidence as seen in this slide. RSRS is introducing all you can learn about review articles, especially systematic reviews and meta-analysis, in just one course. Next slide, please. This is going to be a 7-day course where you will be introduced to the realm of review articles. From topic selection to publication, we'll cover it all. Next slide, Aiza. This is the list of topics that we are going to cover, and the detailed learning objectives are given in the next slide. We have tried our best to incorporate everything about meta-analysis and systematic reviews in just one course with minimal fee. So I invite all of you to join this course, since it will prove very beneficial to you if you want to publish your own review articles. We have very limited slots available, so please secure yours now. And finally, thank you so much for your patient listening. We'll officially begin our webinar now. Over to you, Aiza.
So today is the first webinar. Our speaker for these webinars is Professor Dr. Mujtaba Qadri. Dr. Mujtaba is 4 times US board certified in internal medicine and nephrology. He is the chief editor of Comstock Handbook of Randomized Control Trials and inaugural dean of research and academic development at NUMS, chair medicine at King's Saud bin Abdul Aziz University of Health Sciences, Jeddah. He is the holder of many more esteemed positions. He has published 115 plus abstracts and papers and 2 books. Keeping in view his qualifications, he is perfect for teaching students about randomized control trials. Before handing over formally to Dr. Mujtaba, I'd like to remind everyone to send any questions that you have in the chat box. Now over to you, Dr. Mujtaba.
चलें जी, औज़ुबिल्लाहि मिनश्शैतानिर्रजीम, बिस्मिल्लाहिर्रहमानिर्रहीम. अस्सलाम वालेकुम टू एवरीवन. मेरा नाम मुज्तबा कादरी है और कोशिश करेंगे कि कुछ कॉन्सेप्ट्स उर्दू में बात करें, कुछ कॉन्सेप्ट्स इंग्लिश में करें एज वी गो अलॉन्ग. और सबसे पहले मेरा चैलेंज है कि आप लोगों से आज कुछ ट्राइंग टू मेक टॉपिक विच सम पीपल फाइंड ड्राई, तो उसको थोड़ा सा इंटरेस्टिंग बनाने की कोशिश करते हैं. एंड उसके लिए एक मेरे पास छोटी सी एक स्ट्रेटजी है दैट आई विल ट्राई एंड डू. अच्छा, बाय द वे, आई कांट स्टार्ट माय वीडियो बिकॉज द होस्ट हैज स्टॉप्ड इट. कैन यू लेट मी स्टार्ट माय वीडियो सो दैट आई कैन देन शेयर द स्क्रीन? या कुछ है आप लोगों के पास से? कुछ है? इट सेज अनेबल टू स्टार्ट वीडियो बिकॉज द होस्ट हैज स्टॉप्ड इट. आप लोग अगर मुझे if you don't want to see me, that's fine. I can just share the screen then straight away.
Sir, we have made you co-host. You can try opening your video now.
Okay.
I have made you co-host.
Okay. All right. So
Yes, we can see you now, sir.
okay, great. So let me just share this little bit of a small piece which I do with most of my audience when I do something like this. तो आप लोगों को ये नजर आ रही है स्क्रीन मेरी? Can you?
Yes, sir.
जी, चले. तो फिर I hope everyone can hear me on YouTube also, और इसके अंदर जो है अभी कोई 137 लोग हैं, which is perfect for me to start off this particular series. And अच्छा, किसी ने कहा कि no video, कोई कह रहा है open video please. तो बेटे जरा देखेंगे क्या हो रहा है इधर YouTube पे? Why are people not being able to I think they can all hear me now, right? ठीक है. तो what we will do is कि देखिए, एक तो I just want to give you a quick simple सा introduction to randomized control trials. इसमें नाम से परेशान नहीं होना है, but नाम के अंदर ही सब कुछ है, which is the word randomized. और मैंने आप लोगों को कुछ एक हैंडबुक शेयर की अपनी कॉम्स्टेक वाली, कि उसमें से कुछ चैप्टर्स पहले पढ़ लें. अगर पढ़ लिया है तो वंडरफुल, नहीं पढ़ा है तो आई अंडरस्टैंड, बट एटलीस्ट कल के लिए चैप्टर्स 1, 2, 3 जिसके ऊपर हम आज बात करेंगे, और फिर वो थोड़े से सुबह रिव्यू कर लें जो आज हो जाएगी बात, एंड देन कोशिश करें कि चैप्टर्स 4, 5, 6, 7 एंड 9 वो हैंडबुक ऑफ रैंडमाइज्ड कंट्रोल ट्रायल से you should be able to just go through that quickly, because otherwise 3 दिन में भी randomized control trial समझाना या सीखना would be quite a challenge. तो why do we do research? Research हम इसलिए करते हैं ताकि we try and build up on existing knowledge and कोई answer gaps in knowledge. और एक Zwed Boji साहब थे, उन्होंने कहा कि research is to see what everybody else is seeing, but then ask what nobody else has thought of before. And वही वाली बात है कि आपको कोई चीज अगर समझ में आती है कि इसमें इंटरेस्ट है और ये बड़ी इंपॉर्टेंट है, go and do a literature search and find out if that thing has been answered before. If not, great. If yes, what are the gaps in what has been answered before? And then you can think about a research question which will answer any particular topic of interest. Or not every design has to be at the top of the hierarchy. Systematic reviews and meta-analysis are right at the top, mainly because they put together somebody else's work in terms of high quality randomized control trials or cohort studies, and simply pool the data, and so that one can come up with a better conclusion than what has been previously known about some particular intervention or condition or preventive therapy, as the case may be. So abhi jo hai, when you are looking at trying to do randomized control trials, तो जाहिर है कुछ चीजों के लिए randomized control trials करना मुश्किल है. आप मुझे बताएं, आप में से कोई चैट बॉक्स में लिखे कि अगर if I were to study the effects of soldiers jumping out of a helicopter, and what would be the effects with those who were wearing a parachute and those जिनका parachutes ना खुले, so what kind of a study would be best to answer that? I'm going to wait for somebody to answer that in the chat. हेलीकॉप्टर से लोग छलांग मार रहे हैं, सोल्जर्स, किसी के हेलीकॉप्टर से पैराशूट नहीं खुलते, किसी के खुलते हैं, और उसका इफेक्ट देखना है जमीन पे लगने के बाद. How many survived? What kind of a study would be ethical and you could do that? कोई लिखे चैट में. So let me give you 4 choices. Would it be so a retrospective study, and particularly Arbaz Golabro ne kaha, and particularly it could be a retrospective cohort. ठीक है, तो पहले अगर किसी के साथ हुआ है जिनके obviously जिनके parachutes नहीं खुले, उनमें से कितने survived and कितने did not survive, versus those जिनके parachutes खुले, उनमें से कितने survive and कितने did not survive. But you would be obviously not be able to do a randomized control trial, as Malik Umar Daraz is saying. That would be totally unethical. So जरूरी नहीं है कि हर चीज के लिए RCT कर सकें. Similarly, कुछ चीजों के लिए आप केस कंट्रोल करेंगे, कोई रेयर चीज के लिए, कुछ चीजों के लिए आप cohort studies करेंगे, अगर कोई longitudinal कोई incidence of some study of interest देखना है, लेकिन अगर कोई चीज rare है तो ये retrospective cohort may be the way to go. तो when you do randomized control trials, उसके अंदर basically the whole premise is कि एक तो random assignment होता है subjects का either to an active therapy or to a control, so to speak. And दूसरी चीज होती है कि purely by chance, purely by random distribution, patients may go either to an active limb or to an inert limb or a placebo. और उसके लिए in order to do that, you need to do an informed consent. तो मैं हमेशा ये करता हूँ कि अपने audience के साथ एक उनसे informed consent लेता हूँ ताकि मुझे उनको समझ में आए कि what are the processes involved in that. So I'm going to enroll you people. You are 161 in a study, and I'm going to see how many of you are going to choose to stay in the course and how many are going to choose to have the alternative. Okay, so let's just start. So we are going to be learning initially how to take an informed consent for a randomized control trial through group therapy, and we'll try and do this in about 15 minutes or so. So let me just begin the process. So just bear with me as we go through this. So my name is Dr. Mujtaba Qadri. I'm a facilitator in today's RCT session. And very kindly, the Ravelian research team, which has invited me to be part of this, has invited me to do the study on you. And they have diagnosed you as having ITT-ARIC, which if you have not heard of this diagnosis before, it's an inability to take a research-informed consent. And this, to me, is a potentially serious condition, which will need you all to be actively confined to this session for the next 15 minutes, but with a 2-year follow-up. Over the next 15 minutes, which is this pilot study's duration, I propose to subject you to an interactive therapy session as 2 limbs of an open-label phase 3 randomized control trial, hoping to find a cure for ITT-ARIC. Oh, why can't I go forward? Okay. So informed consent is a permission obtained from patients or study subjects seeking their participation after they achieve an understanding of the relevant study processes and risks involved. Today, I seek your consent for a proposed group therapy in which you are my potential study subjects, and you purely by chance would be randomized to receive either my group therapy or its alternate. And here I am, your therapist, right in front of you. So the possible benefits of this group therapy could be an improved ability to obtain an informed consent through what we call experiential learning, and while you go through the process. With the probable reduction in symptoms of your ITT-ARIC, and thus possibility of conducting high-quality clinical trials in the future. In my experience to date, 70% of my previous subjects have benefited from this, and I published this in abstract form and presented this in several meetings. However, I clearly state that please do not be under any therapeutic misconception, because of randomization and because of chance, your actual benefit for enrolling in this trial may be none. You may actually have some harms. You may decide not to use at this stage your right to be informed of any possible risks or benefits of undergoing this session, as well as information about any alternates. You are expected to attend this session and to participate in clinical trials over the next 2 years, inshallah. The risks of attending the group therapy and its interactive format include possible anxiety, which could lead to rapid breathing and shallow breathing, and that will blow off your arterial blood PCO2, or partial concentration of carbon dioxide, and you may have respiratory alkalosis, which, to confirm, I may need to take a radial artery prick, and that may be somewhat painful. Hyperventilation, because when you blow up so much carbon dioxide, may cause you to experience numbness around your mouths and muscle twitching. This can occur because of reduction in your serum ionized calcium levels. This may occur in up to 20% of ITT-ARIC subjects in my previous experience. You may also experience severe boredom and restlessness with development of escapist tendencies, and all of you are free to voluntarily withdraw anytime, but this may result in paradoxical exacerbation of ITT-RIC, and I believe this can occur in up to 15% of subjects in this group therapy. Alternatively, you may choose to attend a coffee or tea session running in parallel to this session at the nearest chai khana. Okay. For those randomized to caffeine, the major component of coffee or tea, please be aware that if relevant, this is classified as pregnancy category C, which could be harmful to fetus. The possible benefits of the coffee session include a state of increased arousal and wakefulness, which may help in your subsequent participation in my study, with a possible similar reduction in symptoms of your ITT-ARIC, which, to remind you, is an inability to take a research-informed consent. This is likely to occur in up to 50% of subjects electing this format, but again, published literature reports are variable. The risk involved in undertaking a coffee session at your nearest chai khana includes possible flight of ideas and paranoia about the rest of our workshop, with potential harmful effects on your ability to take a proper research-informed consent or to subsequently perform high-quality clinical trials. And this, in my experience, may occur in up to 30% of coffee consumers. The more severe adverse effect after having coffee may be the induction of severe polyuria, or increased amount and frequency of urine. In one randomized controlled trial, during the first day on study medications, patients taking caffeine versus placebo voided a mean of 7.848 times versus 6.4 in a 24-hour period, respectively, and the p-value was less than 0.05. The p should not have the EE in front of it, but I call it p because of a specific reason. This in turn may have a disruptive effect on the morale of the rest of the participants of today's session and seed a possible online TikTok dharna. If you choose not to participate in this therapy session or subsequent ones, I will surely be somewhat heartbroken, but I'm used to it at this age of mine. But the Ravelian team of our hosts under under Ibad Rahman and company, Aiza and company, but they will not hold it against you and continue to deliver the remainder of our workshop, inshallah. Even if your attendance for the follow-up sessions falls below 75%, it shall not affect your chances of receiving future Ravelian research grants if Professor Umar approves it, if you still come up with your IRB-approved ethically and scientifically sound protocols. Only myself as principal investigator and the sub-investigators shall have access to the relevant aspects of your records through coded sequences determined by your information technology department, and all information shall be kept confidential, inclusive of your dates of birth and current body weights. Each one of you is entitled to 2 tea and coffee breaks with snacks for completing this session, as well as on each subsequent session, totally funded by your parents or guardians. I can't send you money through this YouTube channel. However, if you attend less than 7 minutes of this and each subsequent session, you shall have access only to tea, but no snack each time. For any trial-related injury, we shall compensate you by providing immediate first aid and any required additional tests or treatment. If any new information emerges during the next 2 years of follow-up regarding this group therapy, our team shall send timely alerts through notifications and emails. The study contact personnel, you can email me here. If Rawalpindi Medical University higher administration disbands this workshop, we may be forced to terminate your study participation before completing the anticipated required follow-up period. In case the Data Safety Monitoring Board finds it unethical to continue to randomize you to group therapy versus tea or coffee and recommend study termination, we may have to stop these sessions. Your withdrawal of active support, poor attendance, and subsequent dampening effect on my morale may be another reason for terminating this study. Our target population or sample size is the entire cohort of researchers enrolled in the Ravelian Research Workshop. And the required sample size is at least 80 participants. I see, mashallah, you're 182 right now, accounting for attrition of 10%, secondary to either severe boredom or you have to go and pass water. With a two-sided alpha of 5%, a power of 90%, estimated using a time to event survival analysis and hazard ratio of 0.7 with a 95% confidence interval between 0.5 to 0.90 for possible benefits. You may agree to participate in our therapy session by signing in on this session or by a show of hands in the chat box. This will be considered to be your, what do you think this is? EX ... consent. Can anybody write what kind of consent this will be? Or can anybody mention what kind of consent it will be? It's called an express consent. कोई चीज express करने के लिए, ठीक है? If you choose not to do the above, your continued presence online and subsequently will be assumed to be an, anyone? It will be an implied consent. If you prefer to delegate the Institutional Review Board Committee to make this decision on your behalf, we shall proceed with our sessions considering this to be a case of a substituted consent. Since we do not have much time, people are getting all excited and antsy online. So to seek your written informed consent, and because of the serious nature of this ITTRIC and the need to together bring your very own student research community, we propose to continue to deliver these sessions based on the extension doctrine. For some of you, especially the ones who are getting very excited online, we have detected severe signs of ITTRIC. And this may be hazardous to your express goals of becoming good researchers. This requires that we deliver these sessions to you as a medical. Can anyone tell me? Emergency, bypassing usual processes. For the rest of you, if your leadership is felt not to be supportive of these therapy sessions, in your best interest, we may have to resort to seeking a court order. Before we obtain your informed consent, I assume that you're participating in this session free from coercion and unfair persuasion, that is totally voluntarily and with full autonomy, that you have the full mental ability to consent to participate, that is, you have capacity, that you have been given full information on the elements of these therapy sessions, that is, now you have disclosure, that you comprehend the information given and appreciates its relevance to your needs, that is, now you have understanding, and that you are above 18 so that I don't have to take your assent and your consent would be sufficient. And those who consent to undergo our proposed group therapy now that we'll start with the RCTs and over the next 2 days, please raise your right hand in the chat. And those of you who wish to have the coffee alternative, please proceed to the nearest chai khana now. And I believe that by authorizing me to go ahead with the therapy plan, you have now made an informed decision. And this consent shall remain valid for the next 2 days. So congratulations. As we start off this course, we have taken you through an informed consent process, which should take at least a day, because when you explain it to patients that they're going to be enrolled in a study, you can't just hurry them up. But then you have also gone through 20 essential elements of research-informed consent, including the 8 core components. So each time you do a research-informed consent for a clinical trial or randomized controlled trial, you have to mention that it involves research, what is the purpose of trial, what are the experimental aspects, and that you may be assigned by random chance alone to an active limb versus a placebo. The treatment probability of randomization, is it 1:1 or 1:3 to active therapy versus control? What are the alternates and what are the risks and benefits of any alternates? What will be the procedures involved, and duration and length of participation, risk inconvenience for any embryo or child. Thank you. I'm getting lots of raised hands. God bless you all. Expected benefits and the concept of therapeutic misconception, subjects and responsibilities, that the fact that you are voluntarily to leave at any time. I have to respect your confidentiality. Who has access to your record besides myself? Maybe just a research team, core research team. What is the anticipated prorated payments I have to pay you, and the anticipated expenses in this trial? If you have any injury, what happens for trial-related injury? How do I compensate you? And if there's any new timely information, how do I present it to you? Who are the study contact personnel? What is the reason for subject termination, and number of subjects in the study? So I'm going to just end this particular portion. It was just to break the ice. I hope you appreciated that I could have made it very, very boring by going straight to my presentation, which I'm now sharing with you, because this involves some heavy concepts that we will try and learn today before we actually go through the methodology tomorrow. But we will be reviewing a particular article from the New England Journal of Medicine about the use of DAPA, which is a drug called an SGLT2 inhibitor. Don't think about it right now. I'll explain what it means. In delaying progression of chronic kidney disease, published in the New England Journal of Medicine. And we will try and learn our concepts of that article by looking at some of those key things that they describe in that article. And I hope by the time we are done with this course and your quiz, you will feel familiar with at least reading a randomized control trial, and you will get motivated enough, inshallah, to then subsequently do one. So let's go ahead and start. I think I will go ahead and start with your RCT workshop specifically. And so today, the concept that we are going to be discussing are right here. So take a look at this, the evidence hierarchy, which means the level of evidence that we use in order to answer a research question or to actually use evidence in practice. That's important. Excuse me. Then we will look at what analytical studies where do the RCTs come in. For the analytical studies, we will try and see the spectrum of translational research. How is a new drug formed? Where does it start? Why does it take 10 to 20 years before something which is a molecule and has been extracted from plants, perhaps, then takes 10 to 20 years before it forms a drug and that comes in the market? And what would have happened if in the COVID times your vaccines didn't come out on an ultra fast pace. Some of us may not have been around today. We would have been dead in our graves, wondering why somebody didn't do research in time, and some of us could have been alive. So, so these are all, everything comes in because somebody out there, some human volunteers, some human patients are risking themselves to be enrolled in high quality trials, so that when some kind of a positive effect is shown, then you have more convincing evidence that that drug, if it is safe, can be used in clinical practice. And that is how all of us take whatever medicines we take for granted, like any medicine. Think of Cefspan, think of azithromycin, think of Augmentin, think of furosemide or Lasix, think of any drug in the world. All of that has come into the market through a process, and a lot of its competitors have fallen off because the clinical trials that were done were either not of good quality or the drug simply was unsafe. So this is why we need to understand why clinical trials are done, and all these vaccine trials with the COVID and all that, they are all clinical trials. I think maybe some of you may be familiar if you've seen it being done in your centers. Some of you may not be familiar, but I will go through that. So we will go through all these concepts. We will review the types of clinical trials. Then we'll look at the concept of bias, contamination, confounding, core interventions, validity, what kind of interventions, primary, secondary, what about the endpoints, primary endpoints, secondary endpoints, whom to include in a study, whom to exclude in a study, how do you recruit patients, how do you calculate the sample size, what is this data safety monitoring board, what is interim analysis, missing data, study question and hypothesis testing, and the ethics of research. So today we will just simply concentrate on these concepts. And then if you are good students and you are motivated and you read the handbook that I've sent out to you through Ibad earlier on, at least read chapters 4, 5, 6, 7, and 9 for tomorrow. Then you may understand some of what we are talking about. So let's just look at the evidence pyramid again. So Brian Haynes is a guru, and Gordon Guyatt is the father of evidence-based medicine. David Sackett was the original, I guess, grandfather. He's dead. Gordon Guyatt is a dear friend and mentor of mine as well. And I've learned a lot from him regarding evidence-based medicine and how to interpret randomized controlled trials and its quality since about 15, 20 years. And he's come a couple of times to Pakistan in 2007 and 9. And we did some workshops with Gordon Guyatt on evidence-based medicine at Shifa College of Medicine when I used to be there. So, so I think Ibad, somebody is saying that maybe you need to share that handbook, the handbook of randomized controlled trials. Please send it in both the chat boxes so that at least they can go through chapters 1, 2, 3 after this session, and 4, 5, 6, 7, and 9 for tomorrow. Okay, so if I just, I hope all of you can see this slide. So if you're just looking at right at the bottom, the background information and the expert opinion. So you know what experts? I call myself an expert, and I call myself an expert because I believe in Niels Bohr's definition, the famous physicist. He used to say that an expert is somebody who has made all the mistakes that can be made in a very narrow field over a long period of time. So that's all we experts, so-called experts, are. And we can mislead you, we can lead or misguide you, and don't bank on entirely what we say on rounds, because we need to be putting that information, expert opinion or expertise, along with best evidence and patient values and preferences in order to provide the best evidence-based care to your patients. So when you're looking at the hierarchy, these observational studies, case control studies, so you start off with case reports where something unusual is there, you have not seen it before, and you basically report it and like a history and physical, and then discuss the case and say, oh, only 1 or 2 have been described. So still remain case reports. till somebody describes a series of the similar cases, and that becomes case series. And they're pretty good in trying to tell the world that these cases are coming up. For instance, in COVID, so many new things happened in different systems that everybody could write case report about how it affected the mind, how it affected the kidney, how it affected the liver, how it affected the gut, how it affected the, I mean, like at the molecular level. So people can do so many different things in case reports and case series. But then if something is rare, you do case control studies where you already have a case and you select a match control and see what are the factors which determine the causation or the odds of that case occurring versus similar factors which were there in controls but did not cause a disease. And you look back in time, and most of case control studies are like that. But when you're looking at things going forward in time, or even though you can do retrospective cohort studies, you have risk factors to an exposed or unexposed group, and you move forward in time and look at outcomes, and you describe outcomes in those who are exposed versus those who are unexposed in terms of relative risks down the line. And that's also pretty good evidence. But then in these studies, you can have, these are observational studies. You're not intervening. You're not actually introducing any experiment of a drug or a behavioral therapy or a medical device. That you can do only with randomized controlled trials. And in randomized controlled trials, we will learn all the steps and all the key concepts as we go along, inshallah. When you put together those, good randomized controlled trials, then there are certain synopses like ACP, American College of Physicians Journal Club. Then if you go up further higher, you get syntheses like the Cochrane database of evidence. When you put the synopsis of syntheses together, you can get things like, this ACP Journal Club or evidence-based medicine or nursing, as the case may be. When you put all that together, you can have the National Guideline Clearinghouse from UK or Up to Date, which is the online text that comes out of Boston every 3-4 months, and it updates all the evidence. So that's how, and then when you, of course, integrate that evidence into electronic health records, then you are talking about systems. So this is the hierarchy, and all the whole effort is because when you want to answer a research question, you simply have a small sample from a group of people who may be accessible to you. And then you have to generalize all the findings to a target population, the general population at large. So you need the best evidence in order to generalize your findings from research. That's why the evidence hierarchy is important to understand. So in general, descriptive studies are simply describing things like your case reports, case series, or just looking at people's backgrounds and just describing what things are and reporting things in terms of simple statistics like mean, mode, median, and bar charts and pie charts and things like that. But when you're looking at analytical studies, the name analytical suggests that you have to use your mind. And when you use your mind, you can either do that in your observational studies, whether it is cross-sectional, but cross-sectional, you don't know whether the chicken or the egg came first. And in case control and cohort, you can have missing data. And we're not talking about that. Somebody else may discuss with you observational designs. But when you're doing experimental studies, these can be randomized based on chance alone. You're assigning groups to active or placebo controls, or non-randomized, where you're not but not by chance are you assigning therapies to either active or control. And there's various kinds of historical controls or concurrent controls, and let's not go into that right now. But today we'll be talking about randomized control trials, key concepts. And here, the first one, which is the placebo control, that is where it is ethical to do placebo-controlled studies. Then you use placebo versus an active drug, and placebo looks, tastes, feels, smells, weighs almost exactly like the original drug, except that it has inert substances in it, nothing else. And so you can use placebo controls versus an active drug if nothing has been shown to work in that particular disease, and there are certain ethics of that. You may choose to have a control group where there's no treatment going on. You may choose to have a control group where higher doses or lower doses of the same drug are being used. And you can choose to have a control group where you have an active therapy. So if something is actually actively working, you cannot randomize people to placebo against an actively useful drug. You have to use an active control. So if you have no treatment of something, then you can use placebo versus an active drug and try and show that the active drug is superior to placebo. But if you already have a standard of care, then you use your active drug against the standard of care, and you can use active drug plus standard of care versus standard of care. And then the types of randomized control trials may be superiority, that you want to show superiority over something, one drug over the other. Equivalence, that both drugs are equivalent, or both behavioral therapies or both devices are equivalent. Or non-inferior, that they are probably not equivalent, but they are not too inferior either. So there's a certain margin below which you may accept that, yes, okay, we can accept 19-20 ka farak, but that 19-20 may at least the one drug which is working at 19 at least has lower side effects, is less expensive. So let's just do a non-inferiority study. And that's how this particular design has come about. So just going to the spectrum of translational research, I mentioned to you that any drug or vaccine has to go through a basic science, and this period takes a good 10 years itself, 8 to 10 years. You take a drug which seems to have some value from plants, perhaps. Let's say you extract a molecule, you purify the molecule, you try and understand its structure, then you try and run computer-based in silico kind of studies, which are known as bioinformatics. So you use your drug structure in computer model simulations and see if it will work at a particular level in the liver or the kidney or the brain or whatever the target is. And then once you're done with that, then you try and use that drug in cell cultures. So you may take some isolated liver cells or isolated kidney cells from animals, and you immortalize them by trying to put them in a certain kind of a culture medium where they continue to replicate, just like cancer cells, for instance. And then you do cell culture and you try and show the same effects in those, and also try to understand any adverse effects. Then once that is done, then you go to small animals like small mice and doves and other things, and you try and do studies on their isolated kidney or liver or other organs, and you do that obviously with a lot of humaneness. You have to be very careful dealing with small animals, and you don't want to create, you don't want to do any kind of things which will hurt them or torture them, or unnecessarily use large number of animals. So there are some ethics of using that. Then you go to perhaps bigger animals. Maybe you go to doves, or you can go to perhaps some studies you'll have to go to primate models like chimpanzees and dogs and other medium-sized or large-sized animals. Once you find an effective dose in that and safety, then you use 1/10 of that dose in human volunteers in phase 1 clinical trials, where about 25 to 100 people will volunteer to be on those studies on that, volunteer to take a dose of the drug. And then you slowly increase the dose till you make sure that the effects are there, but there are no bad side effects. And then you stop if you start seeing too many volunteers developing side effects, and you don't go beyond that dose. So once the people go through these phase 1 trials, then you use larger number of volunteers, maybe 500 to 1,000 volunteers, and do phase 2 studies. So the first studies, phase 1, are safety studies, safety and dosing. In phase 2, you do safety, ongoing safety, and efficacy. Are the drugs actually effective or efficacious rather? And then you do, once you're done with that, then you do clinical trials in thousands of people, up to 10,000 or more, where these randomized control trials come in. And there you are trying to show effectiveness in real life settings, in real diseases that the drug has been made for. And then once the drug is approved after 2 or 3 high quality randomized control trials, then you have to keep the drug under surveillance for 8 to 10 years and report ongoing side effects or any other unusual effects. And eventually, once you start making population level outcome changes or real benefit to society, that is the whole spectrum of translational research. So in other words, you go from the bench to the bedside, to the clinic, to the community, and to the population at large. And you can move backwards and forward between, like it is all translational scientists. Some of them are bench researchers, PhDs, some of them are clinicians, some of them are public health workers, and some of them are policymakers. So this is how the whole science of translational research works. So in a clinical trial, you are comparing the effects of at least 2 different interventions, experimental and a control, in actual human subjects. And that's why the ethics of it, the safety, and the quality of the studies are so important to understand, and they're expensive studies to conduct. They're always prospective. You follow up subjects from a defined moment of their condition, and the interventions are decided by the researcher. And through chance alone, you may allocate people to either an active limb or a placebo. And the studies can be of a preventive nature, diagnostic nature, treatment or therapeutic nature. And the control is the group which is getting inactive therapy, if it's a placebo, or if they're just getting standard of care, if you're comparing standard of care to the new drug. And both the groups are administered treatment in a standardized form, so that you try and see if it actually makes a difference in the outcomes. And if something works, a drug works, or a treatment or an intervention works, you can ascribe causality to that particular treatment. The types of these randomized trials can be usually conventional, which is the parallel group, which we will talk about mostly. There's a crossover, there's an equivalence, factorial, cluster, sequential, and individual N of 1. Let's just look at what a parallel group looks like. So you have a population of interest, and you have a study question, and you have inclusion and exclusion criteria, and you basically address those people through some advertisements that you would like to take part and take an informed consent, like we just went through today. And once we do that, once after they satisfy eligibility criteria, they will be randomized to either the exposed group, where they'll have the active drug, or the unexposed or control group, if it is a simple placebo-controlled study. And you will see an outcome of interest. For instance, if you are looking at a drug which decreases, which improves survival after heart failure, then yeah, I mean, people will either die or they will not die in a certain observation period once they get this new drug. And you will see those who are getting some previous standard treatment, whether after the same period, they will either die or not die. And then you compare the two and see whether you made a difference by doing this randomized control trial. And this is the simplest schema I can come up with for you. But when you're doing a crossover trial, in crossover trial, you use study participants, and after you randomize them by chance to treatment A or treatment B, they will continue to take treatment A for a certain period, and then they will stop, and the drug will come out of the system. And then initially treatment B will be given, then it is stopped, then in this washout period, the drug comes out of their body through urine or bile or whatever, skin or whatever. And then once that is done, then they cross over to the other treatment. So those who receive treatment A will now receive treatment B, and those who receive treatment B will now receive treatment A. And then you compare the effects of those treatments pre and post in the same group. You can compare, or you can compare with each other in the two groups if you're comparing the sequence of which one will be better. This is the crossover RCT. Factorial design, don't get flustered by this particular table. All I'm trying to say is that it's like a 2x2 or a 3x2 table, and you can use various interventions at different levels in various groups. Either you can have 2 active drugs or 1 active drug and 1 passive drug, or you have neither, no active drug, no passive drug, and so different combinations, and you try and find out which group does best in various combinations. So people do that, which is known as a factorial design. We're not going to study that too much. But the simplest types of superiority and equivalence and non-inferiority can be understood by either this particular chart, where 0 is the point of no effect, and these studies which are beyond a certain margin of superiority, this dotted line, if these are superiority studies, this is not entirely superior because it is not crossing the superiority line. Similarly, in equivalence studies, you may have a point of no effect, 0, either superiority or inferiority, and drugs may be either slightly superior or slightly inferior, but do not cross the inferiority or the superiority margin, and these would be considered equivalence studies. In non-inferiority designs, you will have a drug which will be slightly inferior to the original, perhaps, but not inferior than this particular margin. You might be able to see the next slide better, although I can't see it even that well. But if this is a point of no effect, these are clearly superior. These are crossing the line of no effect, so you can't say anything about them. This is inconclusive. And similarly, here in non-inferiority, this is the line of no effect. Studies, drugs which fall here above this inferiority margin, they would be considered non-inferior. And those which cross the point of 0, you can't say anything about that. That would be inconclusive, both this one and this one. And this one which goes below the inferiority margin, again, you cannot make any judgment, but this would be clearly non-inferior. And in this particular last one, this group here and this group here, slightly better, slightly worse, these are considered equivalent. Those which cross the margin of inferiority or margin of superiority will not be considered equivalent studies. So don't get confused by these terms. We will try and see if we can address that as we go along, inshallah. So when you're looking at randomized control trials, somebody already mentioned that sometimes may not be ethical or possible to assign intervention. You can't randomize people to cigarette smoking versus non-cigarette smoking and see whether they develop lung cancer or not. But obviously, you can do case control studies or retrospective cohort studies of smokers and non-smokers, or those with lung cancer and their controls. Similarly, you can't randomly introduce H. pylori in somebody's stomach and then see if they develop ulcers. It would be not ethical or possible. Similarly, if you have a very, very large sample size, you may not be able to do a study. For instance, if something is so rare that you might not even get an outcome during your study period. So you have to decide what is practical before you do a randomized control trial. So one of the concepts that I want you to understand is the concept of bias. It is not the bias that we have with each other or politically or ethnically or otherwise. This is a conscious or unconscious mistake. It's not I wouldn't say it is a deliberate, but it's a unconscious or conscious mistake that the researcher makes in the study design. So if you do not do proper randomization, you will get what is known as a selection bias. I may choose groups which I want them to get better to an active therapy and which I know will not get better to a placebo. So I will bias the outcome. Similarly, what is known as a performance bias, if the subjects know that they are randomized to active therapy versus coffee or placebo, like we went through the informed consent, those who are on active therapy may perform better. This is known as a performance bias. But if I'm blinded, if I don't know which one is active therapy, which one is control, then I will not specifically try and behave differently because I don't know what I've been assigned to. There's something known as a detection bias. So if I want to see the outcome of randomization between drug A and drug B, if I know drug A is active and I am not blinded to it, then as an outcome assessor in the clinic, I may try to see good effects in drug A because I know drug A is active and drug B is not active. So that will bring in a detection bias. If I know that, if you know that you're not on active treatment, you might drop out because you know that you are on a placebo. So this is known as attrition bias. This is where the concept of intention to treat analysis would come in. If I do not register my trials and I don't publish negative results, then I will have what is known as a publication bias. So these biases have to be removed. And that is why we do randomized controlled trials with all the aspects that come in it. So that when we come up with a result, people believe it and they say, okay, this drug study has been done with very valid methods and they have minimized bias and the methodology was very robust. So we should believe the results and we should start taking this drug because it has good effects and has very few side effects and it actually benefits. So you see that's why we are trying to bring in these important concepts. So when we are looking at selection bias, so your study population will not represent the target population. Remember, I said that you take a sample from an accessible population. So remember SAT, sample from an accessible population, and that has to be generalized to the target population at large. You cannot study the entire target population, so you take a sample. So if you're having selection bias, you will not be representative of the target population, but your study population will not be representing them. So the selection of so measures of risks and benefits would not be generalizable to the population at large, and the concept of internal and external validity will be affected. And validity is what you say you measure, you measure. So that is a very important concept, validity and reliability, but we will look at that later on. There are two concepts of contamination and co-intervention, which are important okay, so who's asking? Somebody's asking, isn't performance bias the same as placebo effect then? Yeah, that is exactly what it is, because you could actually, even if you're on placebo, you may just because you're enrolled in a study, you may perform better. So you can sometimes have that kind of a performance bias also. It is also known as a Hawthorne effect. Hawthorne effect is something where you are on a placebo, but you simply because you are in a trial, you perform better than what you would have normally performed when you're not taking any drugs. So that is a little different. But performance bias, when you are not blinded to being on the active or on the control, then you will obviously, you might perform better when you think you're on an active limb. The co-intervention, so people, when you're doing randomized control trials, you want to make sure that whatever is the standard of care, both the groups are getting the same standard of care. The only thing that should differ in the two groups would be the active intervention versus the placebo, or the active intervention versus the second active intervention. The rest of the interventions, co-interventions, need to be equal, so that if causality is expressed, causality is then determined, that causality will simply come in because you have balanced your co-interventions as well. The other concept we need to understand is about confounders and effect modifiers in randomized controlled trials. Confounders can be considered as something so if you have an independent variable which affects in some way a dependent variable, but then you also have a third variable which independently affects the independent variable and independently affects the dependent variable, then it can confound the effect of the independent variable on the dependent variable. And these confounders need to be also balanced, and randomization balances known and unknown confounders in the two groups. But there's something known as effect modifiers, which are things like age and gender that you cannot change. And age and gender that you cannot change, obviously, in a particular trial, then they will have some effects despite the randomization. So you have to then sift out or do adjusted analysis to see what is the effect of these confounders or effect modifiers on your outcome. Internal and external validity, simply internal validity, I would ask you to think of as the internal design of a study. So the randomization, what is known as allocation concealment, which we will discuss tomorrow, inshallah, blinding, intention to treat analysis, all those things that they are done the right way, minimizing bias, that increases your internal validity or robustness of a study. And external validity is what you generalize to the population at large. So if you have a broad inclusion criterion and a narrow exclusion criterion, then obviously you can generalize your study to a broader population at large. If you have a broad exclusion criteria and narrow inclusion criteria, then obviously you cannot generalize your findings to a large population because you excluded so many in your initial sample. So that, if you simply understand that internal and external validity, that's what that means. So you need to balance between external and external validity. Now, my Ibad and team, we started a bit late. So how long do you want me to continue? Another 10 minutes? 10 minutes is fine?
Yes, sir.
Okay. So let's just wind up in 10 minutes today, inshallah. So, you normally need to balance your external and internal validity, and that's how most studies are done. When you're looking at the interventions, like I said, placebo, you can either have a placebo control or an active control. If you already have a standard of care, you cannot randomize people to active therapy versus placebo, because then it would be unethical to not give people active therapy, unless by not giving that active therapy for a little while, it does not make a big difference. Then you can still do placebo-controlled trials. And active controls are when you already have a standard of care, so you need to compare it to that. What do we understand by endpoints? These endpoints are basically the endpoints you seek to determine in a trial. So you normally talk about a primary endpoint and secondary endpoints. The primary endpoint is the main study question you're trying to determine, and your sample size is calculated to answer that. Your secondary endpoints are additional data that you collect because your study is expensive and so many things are measured, but your sample size is not calculated to include the secondary endpoints. So we can only put some value on things which are shown as the primary endpoint. But if secondary endpoints also are positive, that's great. You got lucky. So you have to do a new study to answer that. But if secondary endpoints are negative and you can't show them as outcomes in your study, it doesn't mean anything because you did not calculate your sample size for secondary endpoints and you did what is known as a type 2 error or false negative. And so we will discuss that shortly, inshallah. What are surrogate endpoints? You are trying to so if I say I want to see after 5 years mortality, then that's a hard clinical endpoint. People are either dying or they're not dying. But if I say after 3 years, I want to see ejection fraction and improvement. So yes, ejection fraction can improve in the if your echo shows an ejection fraction improvement from, let's say, 30% to 40%, that's a good 33% improvement from 30% to 40%. So that I may be able to say that, okay, that's a surrogate endpoint. It seems that it will improve mortality also, but I don't have the money or the resources to carry out this study for that long, so I can't take it that long in time. So I'll go with a surrogate endpoint. Composite endpoints, when you know that you have 2 or 3 important outcomes, you try to combine that and you say, okay, instead of one primary endpoint, I will not only look at people dying, but I'll also look at their hospitalizations with heart failure, the number of myocardial infarctions. They're all important, but then they could either be dying or have heart attacks or have a heart failure hospitalizations. And even if any one of them is positive, then my composite endpoint is positive. So this is how a lot of people do studies where they have some important endpoint, but they're not sure if they'll be able to show mortality differences during their study. So this is known as a composite endpoint. When you're looking at recruiting and retaining people in your study, you have to ethically advertise. You have to take things through an institutional review board and ethics committee. And when you advertise that, you have to basically advertise ethically as well. You cannot just say, oh, you're going to benefit from a study. I'm doing an RCT. I'm going to pay you $100 if you enroll in it. No, sorry, you can't do that. You have to say what are the risks of enrollment. The informed consent would be important. And when you recruit people, then you have to keep in touch with them and encourage them to continue. But then if they want to withdraw at any time, like we did in our informed consent, you can withdraw at any time. And so they need to know all the personnel who are involved in the study so that you are retained in the study. Key RCT stats, I think the number important to know how many arm study it is, 1:1, active versus control. There's a concept of minimum clinically significant difference. So you can either make a less than 20% difference in your outcome of interest, a 20% to 50% difference, or a large difference, 50% to 80%. So you decide whether if somebody is given a drug to increase somebody's height after 2 years by 1 millimeter, is that important, or by 2 centimeters is important? Most of you would say 2 centimeters is important. So that's the minimum clinically significant difference that will make a difference to that person's appearance or sense of whatever, confidence levels or whatever you want to do, or to see the efficacy of a drug. So you have to keep the minimum clinical significant difference, which will make an impact. Your ejection fraction improving from 30 to 31% means nothing clinically, but if it improves from 30 to 40%, that's a good clinical significant. That comes from the researcher. That comes in your denominator. So the larger the minimum clinical significant difference, the smaller will be your sample size. The smaller the minimum clinical significant difference, the larger will be your sample size. Alpha is your type 1 error, traditionally 5% we take. Beta is your type 2 error, traditionally 10% we take. These are concepts that I can go through in a table. Power is usually the ability to detect a change when it exists. 80 to 95% is what we like to keep. So power also comes in the numerator. Alpha comes in the numerator. 1 minus beta comes in the numerator. 1 minus beta is actually the power. So if these things, alpha is high, the sample size will have to be increased. Power is high, sample size has to be increased. Minimum clinical significant difference is low, sample size will be high. It is high, sample size would be small. And variance, the more there's standard deviation and other variability in your data, then again, sample size will be increased. And then you do various statistical tests to look at that particular aspect. Confidence interval, 95% confidence interval means that you're 95% confident that whatever you determine in your sample is actually representative of the population at large. That's all it means. Whereas a p-value means that is your result as a is the result as a on the basis of chance alone, and 5% is what we keep at significant levels, 0.05. Anything less than that, we say it's statistically significant. Anything more than that, we say it's statistically not significant. And simply it means that the result came by chance or not. So the probability of rejecting what is known as the null hypothesis is what a p-value is, and we can talk about it again and revisit it tomorrow, inshallah. There are certain important statistical measures like relative risk, which is the ratio of incidence in the intervention group to the intervention in the control group. But absolute risk reduction is control event rate minus experimental event rate, which is very important to understand, and we will talk about it, inshallah. A relative risk reduction is simply a percentage improvement in risk, control event rate minus experimental event rate over control event rate. Don't get bogged down by these stats today. We will take you through that, inshallah, again tomorrow. Okay. Hazard ratio is the ratio of rate at which patients with intervention experience an event to the rate at which patients without an intervention experience the event. And ratio more than 1 means that you are at a greater risk of developing an outcome of interest, and less than 1, you are at a lesser risk compared to the control. Number needed to treat is the number of patients who must be treated for 1 patient to benefit. So it is 1 divided by absolute risk reduction. I sent this out as a quiz question earlier on. We will go through that later on. Don't get bogged down by it. These are several tests, but I just want you to understand that you can either have comparison tests or correlation tests, as the name suggests, or regression tests, which are like your straight line equation or survival analysis. And we will go through that when we go through some of these aspects tomorrow. Survival analysis, we are going to talk about what are known as time to event curves, and I don't want you to get bogged down today about it. But I want you to go and read those chapters so that you understand that this is basically cumulative probability of surviving over time based when you're comparing two different chemotherapy regimens, and you look at 0.5, half of the patient survival as the median survival, and that is a concept that we will be talking about, inshallah. So log rank, when you compare two different survival rates, you use this test, and Cox proportional hazard, the proportional risks that survival entails in the two groups is studied through this particular test. Don't get bogged down by that today. The Data Safety Monitoring Board simply works behind the scenes and looks at data because you are blinded to it, and we should not, and that's the reason they are looking at data so that you're not taking unusual risks, or patients are not benefiting early. You shouldn't be then continuing the trial for too long, or lots of people are having side effects. You shouldn't be continuing trial for too long, but because you are blinded, you don't want to make any serious mistakes or serious errors or take serious risks. So there's a Data Safety Monitoring Board which works at the background. They are allowed to do interim analysis, and when they do interim analysis, they analyze data at 25% of the duration of the trial, 50% or 75%, as the case may be. And only if there is a major difference, they will let you know. Otherwise, you continue with your study totally blinded to all this data. Data may be missing, and when you have missing data, you can either take an average of all the previous data, or you take the last observation and repeat that observation and just put that as a missing data, or you just take the average of all the observations, put it as a mean thing. So this is missing data occurs more in retrospective studies and observational studies, and it's less likely when you have a controlled setting of a randomized controlled trial. You should always register a randomized controlled trial. Our DRAP, Drug Regulatory Authority of Pakistan, or FDA, or any of these other major bodies recommend that you register your trials so that you report the trial results and don't withhold results, because so many volunteers have taken part in it. Even if there's a negative result, one should report that. When you're looking at literature search initially, you do what is known as a PICO. So you look at a population, intervention, comparator, and the outcome over time. And this is how you put your clinical query in PubMed or any of the data research bases. So for instance, you may say population is all patients over the age of 50 with a low ejection fraction following an anterior wall myocardial infarction. Intervention is ACE inhibitor. Comparator is an AR2 blocker, and the outcome is mortality over 2 years. So this is how you try and put your literature search and come up with a hypothesis where you state that either the new intervention will be superior to your older intervention, or equal to it, or non-inferior, and you will normally have a null hypothesis where you will try and say that there will be no difference in the two groups. But then the alternate hypothesis will be there will be a difference in the two groups, which will either be a one-sided hypothesis when you say it will be superior, equivalent, or non-inferior, or a two-sided hypothesis where you simply say it will be different. We can look at that at some point, but let me just have one or two more questions before we wind up for today. One is this thing about finer. So whenever you're doing a research question, always remember the mnemonic finer. So if something is feasible, interesting, novel, ethical, and relevant, then you can go ahead and ask that research question. By feasible, we mean you need to have sufficient time, study design, adequate sample size, and staff. It should be interesting. It should be a novel thing which fills a knowledge gap. it should always be ethical and it should be relevant. So I think, because today was day one, I'm not going to keep pounding you with this. I would like to end here, and we will start off with the background and hypothesis testing and all that, and try and go through this and combine it with the second part tomorrow. So if it's okay with Ibad and team, we'll end here. I can take a few questions if somebody wants, or try and see what I can answer, and we will try and start this off tomorrow, inshallah, at 7:30 sharp, so that we don't miss out anything from final. So we'll start off with this background hypothesis. So I'm going to end right here, and I can take any questions if you like.
Thank you so much for your time today, sir. We have some questions in the chat. Which calculator we should use for RCTs?
Yeah, RCTs, normally you can use SPSS, you can use Stata. Those are obviously statistical softwares, but there are several online calculators that one can use. CliniCalc is a very simple one that you can use, CLINICALC, and you can use that. But normally people usually use the sample size calculators from either SPSS or SAS or Stata or whatever statistical softwares are available, depending on whether you are trying to show an improvement in your hazard ratios or survival curves, or pre and post comparisons in terms of paired t-tests. There are various things that you can use, but the simple answer to that is there are online calculators available. Several of them are there, all free. And simply, once you know that there are one or two groups, what is your effect size? What are you trying to show in terms of improvement? So we will go through a little bit of a small calculation for this particular DAPA CKD trial, inshallah, tomorrow. So you might be able to get some idea of how we are calculating sample size tomorrow. But the simple answer to that is you can use online calculators or one of the statistical software. Yes. Somebody wanted to know about surrogate endpoint and primary and secondary. Okay, so primary and secondary endpoints, the primary endpoint is the primary research question you're trying to answer, and your sample size is calculated for that primary endpoint only. The secondary endpoints are ancillary additional questions you're trying to answer, and since your sample size is not calculated for that, if you get a negative result in the secondary endpoints, you simply may have made what is known as a type 2 error or false negative. So primary endpoint is the primary research question, secondaries are the additional question. Somebody wanted to know about surrogate endpoints. So for instance, if I wanted to see effect of an osteoporosis drug on development of fractures, I could wait for 4 years for somebody to develop fracture or not, or I could simply do bone mineral density in one year or 2 years and look at certain scores, and those will be surrogate endpoints. So not hard clinical endpoints like mortality, like fracture, like heart attacks, but things which fall in the same pathway, but which will shorten the duration of the study are known as secondary endpoints. And my email ID, I think you can take it from Ibad. It is, you can use quadri.mujtaba@gmail.com. Ibad has that. Somebody wanted to know contamination and co-intervention. Simply, contamination is, so I have 2 limbs of a study. You guys are doing it. Some of them are medical students, some of them are residents, and at night, even though I did everything to randomize you to different limbs, at night you chatted with each other on WhatsApp, and you determined, you got to know what are the interventions in the active group versus the placebo group, although you were in the placebo group. So you have contaminated your limb and you have become more aware. So awareness bias will come in the play. So you contaminated your study. So that is important, not to contaminate. You should be totally blind to the other limbs intervention if you're doing a truly blinded study. Co-intervention. So for instance, if I'm doing a study on a new diabetic medication, but if I'm already on some previous basic medications like metformin. The metformin should be in both groups. If I'm on an exercise regimen of 20 minutes exercise 3 times a week, both groups should be on that. So that if I decide to test a new drug versus placebo, I know that the co-interventions were also balanced. Everybody was on the same diet, was on the same exercise regimen. They were on the same metformin. And now I showed a difference because the only difference in the two groups was the active new drug versus placebo. So that's why the co-intervention obviously is an important thing to also balance. Where in Pakistan RCTs are taking place, Bete Heather, Pakistan mein RCTs are taking place everywhere, but are they proper RCTs? I'm not sure how many really know the methodology that well, but so many RCTs were conducted during the COVID times when vaccines and placebos were used on so many different people. So my purpose behind going with these workshops is to help you understand that it's not a joke to take an RCT, undertake an RCT. You have to know the ethical aspects, you have to know the methodological aspects for your study to be appropriate. Otherwise, nobody will publish it, nobody will believe your results. So people are doing RCTs in several places in the country. We were doing RCTs at Shifa College of Medicine, Shifa International Hospital, at Maruf International Hospital, where I currently work. There's a vaccine trial going on, which is an RCT, and a trial is going on in the ICU using a proton pump inhibitor in ventilated patients versus placebo. That's going on. Similarly, at Armed Forces Institutes, oncology and other trials are going on. AFIC, some trials are going on. Similarly, at Shaukat Khanam, cancer trials are going on. At Aga Khan, several different trials are going on. And we in the Pakistan Society of Nephrology are trying to come up with our own indigenous trials. So RCTs are taking place, but are they good quality ones? I can't say for sure, because I've been teaching this for the last few years, and I see lots of gaps. Kindly include the minimum MID, or what was Abdul Qudus? You want to know the minimum clinical difference in clinical trial? I guess that's what you wanted to know. So yes, so minimum clinical difference is basically whatever would be important, minimally important difference. So if I want to show that my drug will improve people's outcome in terms of kidney disease, chronic kidney disease, then I want to show that 20% of my patients over 1 year will not go on dialysis, or 40% will not go on dialysis. But if I say 1% will not go on dialysis, that may not be clinically that important, right? So I need to be able to show a significant difference in outcome, or a reduction in kidney function of about 20%. If I can save people from that, that will be minimally clinically significant difference. But if I say I want to show a 80% improvement in kidney function, it may be impractical. So I have to come up with a practical minimum clinically significant difference in outcome for us to, and that could be measured in terms of relative risk, hazard ratio, t-tests, survival curves, different statistical ways to then determine what will be the minimum clinically significant difference. Somebody wanted to talk about validity. Yes. So if you take a dartboard and you keep hitting near the bullseye, so you are always going to be valid. If you keep hitting the bullseye each time, you're going to be valid and reliable. So reliability means repeatability, and validity means you measure what you say you're measuring. So when you do a trial, you have to have internal validity. That means the trial's methodology has to be robust, and external validity. That means you have enough people included in your study, in your sample, so that you can generalize it to the population at large. So that's the concept of validity. You have to have internal validity, which will come with the very robustness of your randomization, your allocation concealment, your blinding, and your intention to treat analysis. Terms we will come across tomorrow, inshallah, unless you read about it today. And so we will discuss that tomorrow about the internal validity, key concepts. And when you're talking about external validity, that comes in from your broadness of the inclusion criteria. I hope that's okay. Okay. How is sample size calculated for new intervention, Sakina? Usme, basically, you need to know how many limbs are there. एक limb है, दो limbs हैं, और वो एक limb में दूसरे limb में कितना difference है. For instance, मैं कहता हूँ कि मेरे लिम्ब 1 में मेरा जो इवेंट रेट है हार्ट अटैक का वो 15% होगा, और लिम्ब 2 में मेरा इवेंट रेट होगा 10%. तो 15 minus 10, 5 over 15 is 33% difference मैंने दिखाना चाह रहा हूँ. So that is the minimum clinically significant difference that I will put in the denominator in the calculator for effect size. I will put 33%, 0.33 there. And I will put in the two limbs 1:1, one control, one active. Then I will set an alpha, which is type 1 error of 0.05, which is the false positive rate of 5% that is traditionally set. Because you will always make a type 1 error or type 2 error because your sample is not the entire population. You are simply taking a sample from an accessible population. So you may have some false positive inferences and some false negative inferences. The false positive rate is known as alpha or type 1 error. The false negative rate is known as beta or type 2 error. And beta is traditionally kept at around 10% or 20%. 1 minus beta is the power of a study, which is traditionally then kept as 80% or 90%. Power being the ability to detect a change when it exists. And alpha is the type 1 error. So you put your alpha rate, your beta rate, your power rate, and your minimum clinical significant difference, and the number of treatment arms, and the sample size will just come out like magic. All right. So when you're looking at 50% minimum difference, Abdul Quddus, not always, because let's say I want to show a 50% improvement in my height. So if I'm 4 feet tall and you expect me in 2 years to become 6 feet tall and I'm already 14, I think it will be very impractical to put 50%, right? So you see what is possible, what is practical. So if I say I can improve from 4 feet to 4 feet 6 inches at the age of 14 with hormonal therapy, then yeah, that 6 inches addition to 4 feet would mean about a 12% or 13% minimum difference. So you have to decide what will be meaningful. And remember, if you keep something which is very large as your target difference, you may not be able to achieve that particular outcome. If you keep it very small, you will achieve it, but then because it is in the denominator, your sample size will have to be much larger for people to believe your results. Okay. So I think that's it, I guess. और कोई questions हैं किसी के? नहीं है तो even if we have 86 online at the end of this session, I am delighted, because it means that inshallah, if we can retain some of you for tomorrow also, and if we come out with a little bit of a concept and quiz tak, so hopefully you'll find कि कुछ ना कुछ चीजें समझ में आने लगी हैं. It's not something to understand or learn in one day, but उसकी वो किताब मैंने इसलिए आप लोगों को भेजी है. प्लीज इबाद एंड कंपनी, आप लोग आयजा, आप लोग शेयर करें वो इन लोगों के साथ. इनके व्हाट्सएप ग्रुप में वो रैंडमाइज्ड कंट्रोल ट्रायल्स की मेरी हैंडबुक, और उसके अंदर इतने ब्यूटीफुली छोटे छोटे चैप्टर्स मेरी टीम ने लिखे हैं कि यू शुड बी एबल टू रीड इट. मतलब 15-20 मिनट में पढ़ सकते हैं एक चैप्टर, इतना लंबे है भी नहीं. तो आप अगर वो पढ़ लेंगे ना, तो आज की बातें शायद कुछ समझ में आएंगी. कल जो हम करेंगे यहाँ से स्टार्टिंग फ्रॉम बैकग्राउंड हाइपोथेसिस, वो भी समझ में आएगा. ठीक है, shall we wind up now?
Thank you so much, sir. It was a very informative session. Before officially ending the webinar, I'd like to invite Dr. Afifa, Chief Coordinator, RSRS, for her closing remarks.
Assalamu alaikum, sir. Sir, मैं डॉक्टर अफीफा हूँ. I happen to be one of your associate also when you were at Shifa College of Medicine, and I don't know if I can switch on my video. And I met you recently at PSP Con in Peshawar.
जी बिल्कुल.
Sir, I am the faculty in charge of Ravelian Students Research Society here at Rawalpindi Medical University, and I'm serving as Assistant Professor Community Medicine here also. So I'm really delighted. I'm really very grateful कि आपने time spare किया, and when we started disseminating कि आप ये workshop करा रहे हैं, so we received such an overwhelming response, not only from students but consultants, professors, postgraduate trainees, all were really very excited to attend this, and we are really grateful on behalf of RMU and on behalf of RSRS. I'm really grateful कि आपने time निकाला, और the session was really very informative, and we really look forward to further collaborations with you also.
Inshallah, I'll be delighted to help out. And Kal is in concepts, inshallah, let's start building up on the concepts today and newer concept and quiz में inshallah, hopefully जो चीजें unclear थी वो हम आपस में साथ ही collectively learn करके इसको further clarify करेंगे. ठीक है? That's our
Inshallah.
strategy. Inshallah. Thank you. We'll see you tomorrow.
Thank you so much, sir. Thank you.
Assalamu alaikum.
Thank you, ma'am. Thank you, sir. I'd like to remind everyone about your course on systematic review and meta-analysis. Last date to apply is June 4th. Thank you. Allah Hafiz.
Allah Hafiz.